Hello and welcome to my channel Sam Biology Class. Friends, today we will discuss about primer working. Before that, I request all of my friends, please subscribe my YouTube channel and also watch, like and share my videos with your friends. Primer working is a technique that uses a series of Sanger sequencing reactions to determine the sequence of a whole plasmid or PCR product by designing primers to sequence overlapping sections. This technique is useful to sequence moderately long DNA strands, particularly those are between 2000 and 10,000 base pairs long. Primer working is also used to confirm the reference sequence of a known plasmid or a PCR product based on the reference sequence. The first one is sequence discovery service and second one is sequence confirmation service. Automated DNA sequencing machine cannot sequence long DNA strand as a continuous read from start to finish using traditional Sanger's method. To address this limitation, primer working was first proposed in the early 1990s. This technique is an alternative and complementary method to other sequencing strategies such as shotgun sequencing. Primer working is an example of directed sequencing because this process starts from a known sequence and uses new primer to sequence each overlapping section step by step until the entire template has been read. The name primer working comes from the step-by-step process where the end of one DNA sequence read is used to design a new primer in order to sequence the overlapping section of the DNA template. It is like taking a walk with each new primer stepping into the next section. This process is repeated until the entire DNA template has been sequenced. This figure illustrates the basic principles of primer working. This is the DNA template which has to be sequenced. This template has been incorporated into a suitable vector. As already said, in primer working, the long DNA strand is not sequenced as a continuous read. Instead, sequencing is done in overlapping sections by using a series of DNA primers. Primers are short oligonucleotide chains. The first primer is designed based on a known piece of DNA sequence, such as the vector backbone or an antibiotic resistant gene or a specific promoter. In this example, based on this known sequence, first primer was developed and was used to determine the sequence of the first segment of the DNA template by Sanger's method. This constitutes the first cycle of sequencing. So after first cycle, we know the sequence of this segment. Based on its sequence, second primer was developed and was used in the second cycle of sequencing to determine the sequence of the second segment. After second round, as we know the sequence of the second segment, based on it, the third primer was developed and was used in third cycle of sequencing. Here, these two sections are overlapping. Similarly, these two are also overlapping. This is because third primer has been developed based on the sequence of the second segment, and second primer has been developed based on the sequence of the first segment. So in primer working, By using a series of primer, long DNA strands are sequenced in overlapping sections. Friends, this is all about primer working. For more updates, subscribe my YouTube channel, Sam Biology Class.